What do you think life would be like if your neurons experienced turnover just like your skin cells? That is, if they were born and died regularly, like the outer layer of your skin, which has turned over 1,000 times during the course of an average human lifespan. We'd all feel a little bit like Clive Waring, the amnestic patient whose journal is just a series of sentences like, "Now I'm finally awake," with each previous one crossed out. How would we remember anything? How would we learn from our experiences? Would we be able to build skills like language or driving or playing the violin? Probably not. And so, unlike most of the other cells in our bodies, our neurons stick around. Most of them are partners for life. That's why neural development in childhood is so critical. If you don't build those cells properly in the beginning, it's very hard to overcome the disabilities that will result. At the other end of the lifespan, these cells are subject to some of the same effects of aging that deteriorate other parts of our bodies. Comparing performance by people of different ages on many cognitive tasks, it's hard not to wonder whether we reach our intellectual peak in our 20s or 30s and then endure a long, slow, inevitable decline. And that's why, for decades after the first evidence that demonstrated the birth of new brain cells even in adults was published, many neuroscientists refuse to shed the dogma that once we reach adulthood, we only lose neurons. We don't grow new ones. It's not that our brains weren't thought to change. It was known that experience shapes the very structure of our brain cells. That's one way in which learning becomes embedded into our biology. The connections between neurons get strengthened or weakened as memories are formed and forgotten. The amount of real estate that our cortex assigns to different functions changes depending on how we spend our time. And even the synchronization of millions of cells chanting together in a chorus is malleable with experience. We call this malleability neuroplasticity, which refers to the long-lasting changes that a brain can undergo throughout the course of our lives. We use the term plastic because, like plastic, the brain can be shaped into 1 billion different forms, giving each person who ever lived a unique mind. And as we lay down these experiences, we want to hold on to them as long as possible, of course. It's helpful to remember who behaved as a friend or a foe in the past, even if it's been years since your last encounter. These long-term memories are part of the reason why neuroscientists were so reluctant to accept the data overturning the no new neurons dogma. Throughout evolution, our ancestors' brains changed dramatically over the course of a few million years as they began to use tools, live in larger and larger social groups, cook food, and so on. It made sense that the price that we pay for remembering the past and learning how to live together in relative harmony, ultimately building civilization, was the absence of neurogenesis, the birth of new neurons, in adulthood. As you can imagine, neuroplasticity, an ever-changing brain, arguably captures our own experience of our minds much more accurately than the idea of a brain being a computer or other machine whose parts don't change. Thoughts come and go, we learn and we forget, we improve and we regress. But of course, there are some functions that seem to be more amenable to change than others. It's easy to learn that someone has changed her phone number because she moved across the country. It's hard to remember what that new phone number is when we've dialed the old one thousands of times. And as you're probably well aware, there are times in our lives when our brains are more plastic than at other times. It's much easier to learn a new language if we've been exposed to it before the age of 5, when our brains are in what's called the critical period for language acquisition. But it's not impossible to learn and even master a language in adulthood. It just requires more effort. And admittedly, the process by which a new language is acquired is a bit different in adulthood compared with how it works in childhood. To understand this difference and to begin to grasp the nature of neuroplasticity, Let's look at what's going on inside a toddler's brain. When I watch my toddler jump from one activity to the next in mere minutes, or when I try to keep his attention on some task like brushing his teeth or putting on pants, I sometimes get exasperated by his inability to focus for any length of time. His attention span is so short. But actually, a more accurate description from his perspective is that his attention is in fact almost infinite. He's paying attention to almost everything, which is why he gets distracted so easily. His brain is awash with a protein called brain-derived neurotrophic factor, or BDNF for short. BDNF plays an important role early in our lives as it encourages the growth of new neurons and the formation of new synapses so that cells can communicate with each other. It's prevalent during critical periods in which neurons are finding their places in the brain, a time of extensive neuroplasticity. These critical periods are why children who are not exposed to certain types of stimuli, for example, kids who are deaf or visually impaired or aren't spoken to, must then endure a lifetime of disability. If a child doesn't get the right type of stimulation, his cells won't organize themselves in such a way that they can process that information in the same way as in a healthy person later in life. But the work of BDNF isn't finished once a child reaches adulthood. Even in adulthood, we now know that we do grow new neurons in select parts of our brain, regions involved in long-term memory. So the idea that once you reach your 20s, you're stuck with the brain cells that you've got is a myth. But note that neurogenesis doesn't happen all over the brain. As we speculated at the top of the lecture, that would be a disaster. Instead, we have found adult neurogenesis in only 2 parts of the human brain so far, both of which are involved in creating memories. In one region of the dentate gyrus, it's a very specific part of the medial temporal lobe involved in the rapid formation of new long-term memories, and in the striatum, which plays a role in planning actions, reinforcement, motivation, and decision-making. In the striatum, the cells born in adulthood are interneurons exclusively. That is, they don't connect to cells outside of their immediate vicinity. And in the medial temporal lobe, the new neurons turn into granule cells, small, tightly packed cells whose reach is also limited locally. We think, therefore, that adult neurogenesis enables us to encode new memories, giving us the opportunity to learn throughout our lives, but it doesn't interfere with connections that we've made from previous experiences. How do we know that we grow new neurons? Well, some of the first evidence came from bird brains, songbirds to be precise. In the 1980s, scientists found out that canaries grow new neurons in the hippocampus, the avian analog of our own memory powerhouse, when they're learning new songs. And the more complex the repertoire, the larger their hippocampus. Males, for example, have larger hippocampal volumes than females, whose songs are less complex. Males, you see, have to croon to seduce their mates. And canaries aren't the only birds in whom we see learning-related neurogenesis in the hippocampus. The black-capped chickadee can hide up to 10,000 bits of food in different places over the course of the autumn. And in order to remember where each snack is located, their hippocampi grow in size in the fall and winter, shrinking back down in spring and summer when food is easy to find. Why, you might ask, do their brains shrink down again? Wouldn't it be helpful to keep up the volume? Neuroscientists who dismiss these results as limited to birds argue that the birds had small brains to begin with. They have to keep themselves light so that they can fly. And so the adaptation of seasonal memory storage specific neurogenesis made sense. But our brains are big, and we don't have to worry so much about how much they weigh. So there's no reason to think that such a power might extend to humans. After the discovery in songbirds, however, other non-flying animals were shown to possess the ability to grow new brain cells. Neurogenesis in adulthood was reconfirmed in rodents, our relatively close mammalian cousins, genetically speaking at least. You see, decades earlier, in the 1960s, evidence for the birth of new brain cells in adult mammals had already been published, and in prestigious journals too. But the scientific establishment refused to take the data seriously, at the peril of the careers of those who continued to study the phenomenon. But once the songbird story came out, interest in the topic began to spread. From rodents, we've learned that the speed at which these new neurons are born can vary. Rich environments that provide a lot of stimulation can increase neurogenesis. Memory tasks and socializing and even simple play can too. And rats that got a lot of exercise, say by running on a wheel in one study, doubled their neurogenesis rate compared to sedentary ones who weren't provided with a running wheel. What's more, it's not enough, of course, to just grow the new neurons. They also have to survive. Animals who were then given the chance to learn a new skill, like balancing on a moving tube, for example, were left with a greater proportion of surviving new cells compared with a control group, who experienced neurogenesis but then weren't given the opportunity to put those cells to action by learning a new skill. And that's one of the most interesting findings from this literature. The new neurons need to be integrated into adult brains. And that doesn't happen if the animals aren't challenged. Once neurogenesis was induced, rats that were given a task that was too easy were left with fewer surviving cells compared with rats given a hard task to work on. And those animals who couldn't learn the hard task also didn't have as many remaining new cells as those who mastered it. Maybe they weren't trying hard enough. These findings might give us a clue as to the mechanism by which we can keep our brains looking young. We've talked about how challenging the brain in adulthood seems to protect it from cognitive decline. Maybe adult neurogenesis is part of the story here. If you don't use those new cells by learning a new skill, for example, they don't get integrated into your brain. But learning a new skill might be a way to ensure that you don't waste any newborn neurons. Despite the large similarity between our brains and those of rats, there were still prominent neuroscientists who refused to believe that adult neurogenesis was a human thing too. Then, at the turn of the 21st century, a collaboration between scientists in Sweden and San Diego finally demonstrated the phenomenon in our own species. The authors had learned that the same dye used to track neurogenesis in rodents had been injected in certain cancer patients over the previous decades. They then got access to their brain tissue post-mortem and found the same evidence of the birth of new cells throughout the lifespan in these people. But more questions remained. And that's when the science gets really exciting, when a new discovery leads to more and better questions. Now the field wondered what role neurogenesis plays in the human mind, what circumstances affect its rate, and whether we can treat devastating brain diseases with neuronal stem cells. Since 2013, a series of studies have been published that take advantage of the fact that during the Cold War, nuclear bombs were tested, leaving an increase in the atmosphere in the levels of one radioactive isotope of carbon, carbon-14, and therefore in our cells, including our neurons. So scientists can figure out how old any cell is by measuring the amount of the isotope in the cell's DNA. Using this method, neuroscientists have learned that not only is neurogenesis a common feature of the adult brain, but there are important differences between neurogenesis in rodents and in humans. In the mammalian brains that we've studied, the growth of new brain cells is restricted so far. We've found that in rodents, these neurons end up either in the olfactory bulb, responsible for smell, or the dentate gyrus, a subregion of the hippocampus involved in learning and memory. And as we've discussed, the neurons that are born are fairly small and make connections only with neighboring neurons. They stay local. That makes sense, since integrating new cells into complicated circuits might be a recipe for disaster. So, both rodents and humans grow new neurons in the hippocampus. And the other place where new neuron growth begins is called the subventricular zone in the lateral ventricle. In rodents, These neurons then migrate to the olfactory bulb, likely because the sense of smell is of critical importance to a rat. But in humans, those cells migrate to a part of the brain called the striatum, responsible for coordinating movement and critical for long-term learning of motor and other skills, what's called procedural memory. One more really interesting difference between rat and human neurogenesis goes exactly against the logic that some neuroscientists clung to in justifying their rejection of the initial findings. The idea that humans can't possibly grow neurons because we can remember things from our very remote past. The idea was that if memories are stored in neural networks, those neurons need to remain intact for us to be able to retrieve old memories. If there was a turnover of neurons the way there is of skin cells, wouldn't we just forget our past? Apparently not. At least not in the dentate gyrus. In rats, the proportion of new neurons in the region doesn't seem to ever exceed about 10 to 20%. But in humans, by about age 50, the majority of neurons in the dentate gyrus have turned over. Remarkable. How is it possible that we can still remember old memories then? Well, there's growing evidence that the dentate gyrus may play a bigger role in encoding new memories than in retrieving old ones. In fact, rats with dentate gyrus lesions have trouble learning new things, but not remembering old ones. Lesion a different part of the hippocampus, one in which there isn't adult neurogenesis, and you get the opposite pattern. The animals can learn new things, but they can't remember the old ones. That would make sense. We'd need the new cells in the dentate gyrus to form new memories, but then those memories would be ultimately stored elsewhere, and we wouldn't need that region to retrieve them. Instead, baby neurons there would now be available to lay down a new set of memories, leaving us with the ability to learn new things until the day we die. We now know that decreased adult neurogenesis plays a role in neurodegenerative diseases like Alzheimer's disease, and perhaps could be used in the treatment of Parkinson's disease, where we see a loss of a specific type of cell. It's also been implicated in depression and schizophrenia, and as research continues, I'll bet that we'll find many more conditions in which neurogenesis is affected. At the beginning of the lecture, we talked about the toddler brain and how it's awash in brain-derived neurotrophic factor, or BDNF, and that this hormone affects neuroplasticity. Well, there's a connection between BDNF and neurogenesis even in adulthood. You can actually influence the amount of BDNF floating around in your brain and the growth of new neurons with a fairly simple and cheap treatment: exercise. Several studies have shown that regular exercise can lead to a 3-fold increase in BDNF levels. Even a single session can make a difference. Exercise is effective at ramping up neurogenesis and keeping those cells integrated in the brain. And while exercise can increase adult neurogenesis, stress can impair it. In animal models of depression, neurogenesis decreases under experimental conditions where the animals are placed under stress. In these same experiments, administering antidepressant medications to the stressed animals has been shown to restore neurogenesis to healthy rates. There are other examples of people who successfully increase the volume of specific parts of their brains by training or learning a new set of skills. Not necessarily by growing new cells, but by increasing the connections between them. A Swedish neuroimaging study, for example, tracked the changes in brain volumes in a group of army recruits learning Arabic, Russian, or Dari. They compared these changes with those in medical students who were also studying intensively, though the content of their learning was very different. By the end of the study, the Army recruits showed larger volumes in brain regions that are involved in processing language, as well as our friend the hippocampus, which is necessary for new long-term memories for facts and events, compared with the medical students who didn't show measurable changes in brain size. And these changes correlated with how well the recruits were doing. Those who were mastering the new language and who were trying harder showed greater increases in the hippocampus and language areas compared with recruits who were struggling or not putting in as much effort. These results are similar to a now famous study of hippocampus volumes in students training to be black cab taxi drivers in London. The iconic black cabs in London are known for their impressive knowledge of all the streets and landmarks within a 6-mile radius of Charing Cross. Because they're so knowledgeable, they charge a higher fee, and that makes their cabs the most expensive in the city. To get a license to drive a black cab, drivers have to pass a test called the Knowledge. It takes most students 2 years of daily work to pass it. Every day, they spend time in a classroom, and then they get on scooters and race around the city, learning and memorizing the best routes to different places. One seminal early study compared the volume of the hippocampus in black cab drivers with the rest of the population and found a difference. Their hippocampal volumes were indeed different. But this study couldn't address the question of whether the training itself led to the increase or if people with larger volumes were drawn to the job and were more likely to pass. So in a follow-up study, the neuroscientists tracked brain volumes in a group of students studying to pass the knowledge. And sure enough, everyone started off with hippocampal volumes of similar size. And when the study's authors compared the volumes of the students who passed the test with those of students who failed, they again found differences. The training itself, if it was successful, was responsible for hippocampal growth. Even juggling every day for 3 months has been shown to increase the volume of certain parts of the brain, including the hippocampus, in 20-somethings and in 60-somethings. And while the 60-year-olds weren't juggling as well as their younger counterparts with the same amount of training, they still showed increases in brain volume. But once the participants in those experiments stopped juggling, their regions returned to their previous sizes, just like the rats, who, without an effortful task to do, lost a lot of the new neurons that they had grown. I can't talk about neuroplasticity, however, without mentioning music. That's because musical training has long been used to demonstrate plasticity in human brains. There are studies that show that pianists have more of their cortex devoted to processing sensory motor information from the hand than the rest of us, and that musicians in general show measurable brain differences in many regions. But is this nature or nurture? At least one compelling study has shown that it's musical training, and not just genes, that's responsible for many of these differences. Taking a group of 6-year-old public school children in Boston who had no formal music training, scientists randomly assigned them to 2 groups. One group received individual piano lessons every week, and the other got to sing and play drums and bells in a music class at school. The groups were then matched for socioeconomic status. 15 months later, the scientists scanned their brains again and found that the group with individual music lessons showed larger brain volumes in 3 areas: regions controlling motor behavior, hearing, and the corpus callosum, the fiber tract that joins the left and right hemispheres of the brain. So it does seem that musical training can drive brain volume increases in parts of the brain that are engaged by the activity. As we get older, we tend to spend less time learning new things or engaging our brains in effortful ways. And given that this effort is what seems to benefit our cognitive function and our brain structure in measurable ways, our lifestyle choices play a significant role in whether or not we experience cognitive decline with aging. And that experience, in turn, can affect our very attitudes towards longevity. For example, if you could choose, how long would you want to live? I've been asked that question twice now in a room full of people, and both times I was surprised at what the majority of people said they would choose. Unlike me, it seems that most people don't want to live longer than 120 years. The science writer David Ewing Duncan conducted an online survey on this question and found that out of more than 30,000 responses, more than half of respondents agreed that somewhere around 80 is a good enough lifespan. I'm in the top or bottom, depending on your point of view, of the 5% of people who really want to live forever. But perhaps I shouldn't be so surprised, given the hardships that currently accompany aging. If continuing to live just means continuing to suffer and decline, then sure, at some point, enough is enough. And the stigma that's associated with old age underscores the popular belief that things only get worse. Is that true? At least in Britain and Germany, surveys of life satisfaction have shown that it actually steadily increases between age 40 and 70. If you're Australian, then you're likely to find peak life satisfaction around age 80. So maybe many of those Duncan survey respondents were on the right track. But if we manage to stave off physical and mental decline and save enough money to economically sustain an extended retirement, I suspect that most people would push out their desired lifespan, perhaps even past 150. So, how much do we know about the basic biology of aging? Unfortunately, at least in the US, the amount of funding for the scientific study of aging biology pales in comparison with the amount devoted to studying diseases of aging. That's a bit backward, in my opinion. Once we figured out that smoking increases the risk of lung cancer, we instructed people to stop smoking. Now, we can't just tell people to stop aging, but shouldn't we be racing to understand how aging affects our bodies so that we can slow or reverse the process rather than focusing on the vast majority of our resources on the consequences? When it comes to cognition, there are already hints that interventions designed to keep the body healthy, like exercise, have powerful effects in terms of staving off age-related mental decline. But what about those people who seem to be able to stay mentally sharp well into old age? Is there something we can learn from these superagers? There are several labs now studying not only healthy aging, but super healthy aging. People who thrive in old age and who score as well as people who are 20 to 30 years younger. In one study, for example, superagers are defined as individuals over the age of 80 who score as well or better than people who are 50 to 60 years old on a set of cognitive tests. In one study involving around 30 of these people, scientists found that their anterior cingulate cortex, a region of the brain responsible for cognitive control, resolving conflict, keeping up motivation and perseverance, among other executive functions, was not only larger than that of their 80-year-old peers, but also bigger than the average size in middle-aged people. We still have a long way to go before we can make any solid conclusions about what factors combine to support such successful aging. Certainly genes are a factor. For most of us, lifestyle choices have a greater influence on our risk of death before age 80, and genetics seem to account for more of the individual variability in overall health once we reach our 9th decade of life. In one study, superagers are pretty varied. Attorneys, neuroscientists, even one individual who reportedly smokes a pack of cigarettes a day and caps off each evening with a martini. But what they may have in common reminds me a little of our famous Sister Mary from an earlier lecture, at least anecdotally. They have remained active members of their community, socializing regularly. And sure enough, there's one more brain finding in this group that's been reported. They have 3 to 5 times more von Economo neurons in their anterior cingulate cortex than their peers. These large cells are thought to play a role in facilitating social interaction, and they are the topic of our next lecture.